Okay, so welcome to this next video in which we are discussing the Adreno receptors. Okay, so we're currently in the process of looking at the beta adrenoreceptors, which includes the beta 1 adrenoreceptor, the beta 2 adrenoreceptor, and the beta 3 adrenoreceptor. And we've seen that all three of these adrenoreceptors are coupled to the GS heterotrimeric G protein and therefore lead to an increase in the production of cyclic AMP by adenylyl cyclase enzymes, and that cyclic AMP is then capable of activating protein kinase A enzymes, both of type 1 and type 2. Okay, and these catalytic subunits of protein kinase A that are released when the regulatory subunits of protein kinase A bind cyclic AMP and change conformation will then go on to phosphorylate proteins. Okay, so we now want to get into some specifics. So we're going to start off with the beta 1 adrenoreceptor. Now, the beta 1 adrenoreceptor, the most famous location of the beta 1 adrenoreceptor is the heart. And on the heart, what it causes when it is stimulated is it causes an increase in the rate at which the heart beats, so an increase in heart rate, and it also causes an increase in the force of contraction of the heart, so it has a positive inotropic effect. So I want to discuss the positive chronotropic and the positive inotropic effects of the heart, of beta 1 adrenoreceptors with you. And to do this, we need to discuss the anatomy of the heart and the basic physiology. So I'm just going to make sure that we're all up to date. Well, not up to date because it's been known for quite a while, but that we're all familiar with the anatomy and basic physiology of the cardiac cycle. Okay, so let's start by drawing a picture of the heart. Okay, so the heart is a huge sort of circular organ. Okay, and I'm going to start drawing it like so. Okay, and I'm going to leave that bit at the top for the blood vessels to come out. So let's sort of fix the picture up. So we'll have the right atrium here. Okay, then I'll have the interventricular septum here, and then we'll have coming up here the pulmonary trunk, okay, which is coming out of the right ventricle down here. Okay, I'll bring this around a little bit, and then coming out from underneath here, we'll then have the aorta. So I'll have the aorta circling round like so, okay, and of course it will give off its main three branches. Okay, so the brachiocephalic artery, then the left coronary artery, the left common carotid artery, and then the left subclavian artery. Okay, and then it will go down like this. So this is the aortic arch. Then we've got the pulmonary trunk splitting into the two pulmonary arteries here. Okay, and then if I complete this picture up now, so here we then have the aorta coming into the left ventricle here, and then we'll have the left atrium there, okay, and that should be more that sort of angle like so. Okay, and I just want to put some valves in, so we'll have the valves going into the pulmonary trunk and the valves going into the aorta. Right, so here's my picture of the heart. I just want to finish that up. Right, so let's label the different portions up then. So this portion here, which I'm going to colour in blue, I think, is the right atrium here. Okay, and draining into this, you'll have the superior and inferior vena cava. So let's put those in. So here, we'll have the superior vena cava. So this can be the superior vena cava, the SVC. And then from below, we'll have the inferior vena cava draining into it as well. Okay, so this is the inferior vena cava, the IVC. Okay, here we have the right atrium, which I'll just denote as RA for right atrium. Then we have the right ventricle here, which I'll have in orange, I think. Okay, and between the right atrium and the right ventricle, you have the tricuspid valve here. Okay, so this valve here, which I'll just extend and make a little bit more obvious, this is the tricuspid valve. Okay, Then coming out of the right ventricle, you then have the pulmonary trunk here. So this is the blood vessel which takes the deoxygenated blood that's in the right ventricle up to the lungs to be oxygenated. Okay, and at the moment just one blood vessel comes out of the right ventricle, and then it splits into the right pulmonary artery, this one over here, and the left pulmonary artery, which will go to the right and left lungs respectively. But the first one, before it is divided into the right and left pulmonary arteries, is just called the pulmonary trunk. Okay, the blood will go around the lungs and then it will come back in the pulmonary veins, which I'll show here. So there are 4 pulmonary veins, which I'll colour in red, I think, because the blood coming back in the pulmonary veins is actually oxygenated. So these are the only veins of the body where the blood in them is oxygenated. Now there are 4 pulmonary veins, 2 from each lung. So there's 2 from the left lung over here, and then 2 from the right. 2 from the left lung over here, and 2 from the right lung over here. I'm getting my right and left mixed up. Sorry about that. Okay, so here this is the left atrium, the LA. Then it goes into the left ventricle here. So let's colour these bits in differently. We'll have the left ventricle in purple here. Okay, we'll have the left atrium in pink at the top there. Okay, that pink is supposed to be slightly different to this purple here, but it doesn't look that different, I'll give it that. And then we've got the aorta coming off in green here. Okay, and giving off its first major 3 branches. Of course, the first branches will be the coronary arteries, but they'll be down here. The main 3 branches that then come off the aortic arch are the brachiocephalic artery here. Okay, also sometimes called by surgeons the innominate artery, brachiocephalic artery, which will later split into the right common carotid artery, which will go up to the head, and then the right subclavian artery. This one here is the left common carotid artery. So this is the left common carotid. Okay, and then the next one to come off the aortic arch back here, this is the left subclavian artery. So this is the left subclavian artery. Okay, right. Just a tiny little bit more anatomy. The names of these valves here, okay? So these are the semilunar valves. Well, both of them are semilunar valves, but this one specifically is going into the aorta that I've pointed to here. So this is the aortic valve. And then we've got the pulmonic valve here. Oh, and we've missed the bicuspid or the mitral valve as well, so we'll do that one in a moment. Okay, so this is the pulmonic valve here. And then finally, between the left atrium here and the left ventricle, we've also got a little valve here. If I can just sort of get this squished in here. You can't really see it, but it is there, okay? And this is known as the bicuspid or the mitral valve. So bicuspid / mitral valve. Okay, and both the tricuspid valve between the right atrium and the right ventricle and the bicuspid or mitral valve between the left atrium and the left ventricle are considered the atrioventricular valves because they're between atria and ventricles. Okay, right. So, let's now just have a brief revision of the cardiac cycle. Okay, so basically it starts with blood coming into the right atrium and the left atrium from either the peripheral body, which it's coming in through the superior vena cava and inferior vena cava into the right atrium, or from the lungs through these pulmonary veins here. So we're getting blood filling into the atria, and some of this will obviously drip down into the ventricles as well. Okay, right, so you're getting the filling of the atria and the ventricles via either the superior vena cava and the inferior vena cava in the case of the right heart, or the pulmonary veins in the case of the left heart. Okay, then what happens is the Sinoatrial Node, which is located up in the atrial muscle, okay, and it just looks like a normal piece of muscle. You would not know that it was different, okay, but it has extremely different electrophysiological properties. This is known as the Sinoatrial Node, and it's going to begin the process of contraction, basically, and it's often abbreviated to the SAN for short. So this here, this is representing the SAN. Basically, what it will trigger is it will trigger a spontaneous action potential, okay? And it's not just one cell, there are multiple cells in a way. They will produce an action potential, and this action potential will propagate down the atria. Okay, so the atria, the wall of the atria, we're talking about the right atrium and the left atrium, they're sort of in contact basically with one another, even though you can't really see it in my picture. Okay, basically all the muscle cells of the atria are electrically connected to one another. Okay, so if this is one little cardiomyocyte, then you've got another little cardiomyocyte here, and they've got special little electrical windows between them called gap junctions that allow an action potential propagating along one of them to then spread through this gap junction here to the next one, and this one will then fire an action potential all the way down it, and then it will contact the next one along, fire an action potential. So basically the action potential propagates down the walls of the atria, and when the action potential hits a cardiomyocyte, that cardiomyocyte will also contract. So basically you're getting this action potential spreading down the wall of the right atrium and the left atrium because they're connected together, the walls at least electrically. Okay, and that will cause contraction of the two atria. Now the action potential cannot propagate from the atrial wall to the ventricle wall. There is an electrical insulation between the wall of the atrium and the wall of the ventricle, known as the septum, basically. Okay, so there's an electrical insulation between the walls of the ventricles and the walls of the atria, so that this action potential that's propagating down the walls of the atrium cannot just jump onto the walls of the ventricle. Okay, there is one little window in this wall which is at the center here, basically. Okay, and this is what's known as the Atrioventricular Node. Okay, so this is called the Atrioventricular Node, and is usually abbreviated to AVN for short. So the Atrioventricular Node, and it's called that because it's between the atria and ventricles, and it's the only window, the only electrical window between the atria and the ventricles. Now, the action potential will get to the atrioventricular node, and the atrioventricular node is very, very, very slow about conducting it through, basically. Okay, in the meantime, what's happening? Well, the atria are now contracting, and they're pushing the blood from the atrial cavities into the right ventricle and the left ventricle. So the right atrium pushes the blood into the right ventricle, and the left atrium pushes the blood into the left ventricle. Okay, so you're getting all of the, well, a lot of the blood ejected from the right atrium and from the left atrium into the ventricles. And once the blood is in, it can't come back out because these valves are one-way valves. So the tricuspid valve here and the mitral or bicuspid valve over here, they're one-way valves. Basically, if you push blood in this direction, they'll open and allow the blood to go through. If you push blood in the other direction, you will push the valve shut and therefore close off that gap between the atrium and the ventricle. Okay, right. So that is atrial systole occurred, basically. So I'll just give you that piece of terminology. So basically, atrial systole means the contraction of the atria. Okay, so we've just had atrial systole occur. Now, basically, we need to look at how the action potential is going to propagate down the ventricles. Now, you might think, well, great, is it just going to come through here and then propagate down the ventricles? But that's not going to be good, and let me explain why. Because if you just let it propagate down the ventricles, it will hit this portion of the ventricle before it will hit this portion of the ventricle. And that's not good, because you're going to get contraction of this portion before you'd get contraction of this portion. So what would happen? you'd get a contractile wave going down, and that would just push the blood down into the base of the heart. That's not a good idea. So what you actually want to do is you want to take the action potential down to the bottom and then unleash it on the muscle from the bottom, so that then the wave can propagate up, pushing all of the blood up and out of the ventricles. And that is indeed what nature has done. Okay, so basically, let me just draw the wall between those two ventricles in a bit more detail. Okay, so I'll draw it now as having a thickness rather than before we were just drawing it without a thickness. So these are the two ventricles basically. This is the right ventricle, this is the left ventricle. Okay, and up here, let's say here, is the Atrioventricular Node. Okay, so I hope you can appreciate that I've just basically taken the bottom portion of this picture basically and made these walls have an actual thickness rather than just being represented as a little line. Okay, right. So the atrioventricular node does not just pass the action potential onto the muscle on the other side of the septum basically. Instead, it passes it on to a specialized conducting system basically. So a bunch of cardiomyocytes that aren't contractile and instead are specialized for conducting action potentials. Okay, so what happens is you conduct it down a little bundle of conduction fibers here that's called the bundle of His, okay? And this sits in the wall between the right ventricle and the left ventricle, often called the interventricular septum, so I'll call it that. So this is the interventricular septum, okay? And basically, the bundle of His is going to conduct the cardiac action potential down through this wall without passing it to the normal contractile muscle cells yet. So it's just going through these special conduction fibers. Okay, so here's the bundle of His. Then what happens is the bundle of His splits into two halves, okay, like so, called the right bundle branch and the left bundle branch. Okay, so this is the right bundle branch here, okay, and then we've also got the left bundle branch, which is going to service the left ventricle. So this is the left bundle branch, okay, and now what's going to happen is they will pass it onto a bunch of fibers which will then go into the wall of the of each ventricle. So these ones are going into the wall of the right ventricle, but there will be fibers over here which go into the wall of the left ventricle, and these fibers will then pass the action potential to the actual contractile muscle fibers, and therefore set the action potential propagating off up the walls of the ventricles. Okay, so these fibers are called the Purkinje fibers. Okay, and then what will happen is the action potential will propagate up the walls of the ventricles, and where the action potential is, you'll be getting contraction, so you'll get a wave of contraction propagating up, and this will push the blood up the ventricles and out of the blood vessels. So the blood from the right ventricle will be pushed out into the pulmonary trunk here and will go to the lungs, and then the blood from the left ventricle will be pushed up into the aorta and go around the body. Okay, that is then called ventricular systole. So when you get contraction of the ventricles, that's known as ventricular systole. Okay, then what will happen is everything will start to relax again. So the atria will relax obviously first. Okay, so the atria will relax, and that's called atrial diastole, and then the ventricles will relax, that's called ventricular diastole, and then whilst you're in full relaxation, that's called cardiac diastole. Okay, so complete cardiac relaxation basically. Okay, and then of course the heart will refill with blood from the inferior and superior vena cava and from the pulmonary veins, and then the sinoatrial node will fire off an action potential again, and the fun occurs all over again. So that's just a brief reminder of the cardiac cycle basically. What I now want to discuss is what causes the spontaneous action potentials that are being generated at the sinoatrial node, because basically this is how we're going to speed up the heart rate, basically by acting on the sinoatrial node. The faster the sinoatrial node fires spontaneous action potentials, the faster the heart beats. And this is where beta 1 adrenoreceptors are going to act. They're going to act on the sinoatrial node to increase heart rate, at least. As far as increasing the force of contraction, that's a change that's going to have to occur in every single contractile cardiomyocyte. We'll come to that in a moment. But we'll start with the increased rate of the heart. Okay, so for this we need to discuss what causes the spontaneous action potential within sinoatrial node cells. Okay, so let's take one of these specialized cells that we have within the sinoatrial node. Okay, so let's say that this is a special sinoatrial node, SAN, cardiomyocyte. Okay, right, and we want to see what do these cardiomyocytes have which is special that allows them to generate spontaneous action potentials. Well, basically the thing that they have is something called an HCN cation channel. Okay, now what does HCN cation channel stand for? So what does the HCN stand for? Well, the HCN stands for the hyperpolarization activated and then cyclic nucleotide gated. So H is for hyperpolarization activated, okay? And then the C is for cyclic nucleotide gated, okay? And then the N is for nucleotide, so the CN is for cyclic nucleotide gated. And then it's cation channel. So the HCN stands for hyperpolarization activated, that should be, cyclic nucleotide gated. So these are the hyperpolarization activated, cyclic nucleotide gated cation channels. And basically, these are special ion channels that you have within the cell membrane of sinoatrial node cardiomyocytes. Okay, right. So let me show you the structure of an HCN cation channel. So here is the plasma membrane, okay, like so, with the HCN channel sitting within it, okay, like so. Now basically these hyperpolarization activated and cyclic nucleotide gated cation channels, they are tetramers. They're made up of 4 separate proteins joined together, and that's not particularly easy to see on the camera, but never mind. It's made up of 4 separate proteins, okay? So it's a tetramer. Again, we have seen many examples of tetramers now. We've seen IP3 receptors. What other ones have we seen? What else did we do? We did the inwardly rectifying potassium channels. Those were tetramers. Did we see any others? I don't know. I don't think so. But there are loads of tetramers, basically. They are all over the place. And here's another tetramer. Okay, so again, same protocol. Let's pull out one of those proteins that makes up the tetramer. So 1/4 of the tetramer. Let's pull out this 1/4 of the tetramer, and let's have a look at its structure, basically. Okay, so I should just say this is the extracellular fluid side, and this is the cytoplasmic side. Or because we are dealing with a cardiomyocyte, you might like to call this sarcoplasm. Okay, so whenever you're dealing with a skeletal or cardiac muscle cell, you have the opportunity to be pretentious and call cytoplasm sarcoplasm. Okay, right. So let's pull out one of these subunits and let's have a look at the structure of it. Okay, so basically they have a structure that's very, very similar to loads and loads of different voltage-gated ion channels. Okay, they have 6 membrane-spanning alpha helices labeled S1, S2, S3, S4, S5. Then they have this P loop and then S6 here as well. Okay, so here is the amino terminus, and then here is the carboxylic acid terminus over here. Okay, like so. So this is the structure of a monomer of the HCN tetramer, basically. What you do is you stick 4 of these together to make an HCN channel. Okay, now, again, it would be beautifully simple if there was just one HCN channel subunit and you just stuck 4 of those together to make an HCN channel. Okay, but it's not that simple. Instead, there are multiple types of HCN subunit. There are multiple genes which code for multiple HCN subunits. Okay, now there are 4 separate HCN subunits which are known. There is the HCN1, HCN2, HCN3, and HCN4. Okay, now basically the next question we have to ask is, how do these assemble together to make tetramers? Well, basically what we know is that they do assemble to make homotetramers. Okay, so all 4 of them do assemble in homotetramers. So, for instance, if you wanted to make a homotetramer of HCN1, what you would do is you'd make 4 HCN1 proteins, and you'd stick them all together, and then you'd have an HCN1 channel, okay, or an HCN1 homotetramer. And you can do that for all of them, basically. There is growing evidence that heterotetramers also exist, where you have different HCN channel subunit types in one HCN channel. Okay, but we definitely know that homotetramers exist. Okay, right. Basically, what we now want to discuss is what these ion channels actually conduct. Well, basically, they are cation channels, so they're going to actually conduct both sodium and potassium when opened, okay? But we'll continue this discussion in the next video.